Hi, everyone. My name is David Schneider. I'm an assistant professor at the Center for Neuroscience at New York University, and I'm excited to be giving this talk virtually today and to be part of the symposium. And today I want to talk to you about some of our work looking at reflections of action, that is movement, and expectation in the mouse auditory cortex. And I want to talk to you today about some ways in which neural activity and neural responses to sounds in the auditory cortex deviate from ways in which we might expect the auditory cortex to respond, given its kind of classical role as a purely auditory center of the brain. And I have this particular image up on my title slide because I think it really exemplifies one of the key ways in which our brains and our auditory system process sounds, right? So there's a picture of this woman in the background shutting a car door. And what I think is really interesting about this movement is this is an extremely mundane movement that each one of us has done thousands of times. And we've never really explicitly, consciously paid attention, or we rarely do, to what the sound of a car door sounds like, but we each know exactly what to expect to hear every time we shut that car door, right? We expect to hear the stereotypical thump. And we know that we expect that because if we ever shut the car door and we hear something different, it instantly captures our attention, right? So if we shut the car door and we hear a clank instead of a thump, for example, right? We instantly turn around and it tells us that we likely got the seatbelt stuck in the door jamb, right? Or even perhaps more strikingly, if we ever shut the car door and we don't hear anything at all, even the omission of an expected sound captures our attention, right? And it probably signals that we didn't shut the car door hard enough, maybe because we're parked on a slope and we didn't really shut it with enough force, right? So even behaviors as simple and mundane as shutting a car door really exemplify the way in which our auditory system pays attention to expected sounds. And one of the key ways in which the brain can expect sounds is because many sounds are the simple acoustic consequences of our actions. And there's complicated versions of that, such as speech or musicianship, but there's also very simple movements that we make for which our brain can expect the consequences, such as shutting a car door, typing on a keyboard, or the sounds of our own footsteps, for example. And my laboratory is really interested in how the brain learns to anticipate the acoustic consequences of our actions, and we tend to study this using simple behaviors. And we spend most of our time as scientists in the auditory cortex, using the mouse as a model organism. So I have a simple drawing here showing the dorsal surface of the mouse cortex with the auditory cortex outlined in blue. And what's interesting about the auditory cortex, particularly in the mouse, but in other organisms as well, is that the auditory cortex receives dense non-sensory inputs from lots of other regions of the brain, including those related to attention, engagement, arousal, memory, and movement. All of these in addition to primary ascending auditory projections. In fact, if you were to rank order the density of projections into the auditory cortex, those coming from the primary auditory thalamus would sit quite low on that list. And so we believe that non-sensory input to the auditory cortex provides a substrate for learning about how the world works, and in particular, learning how to expect the acoustic consequences of our actions. And we think that it does this by coordinating and keeping track of the statistical relationships between the movements we make and the sounds that they produce in order to build, store, and use something that's called an internal model. And an internal model is something that's built up through experience. It's usually subconscious, and it describes how the world's going to change as we interact with it. And the idea is that we have all sorts of internal models in our brain, and we're constantly using these internal models to build little simulations of the world about what's going to happen next based on our previous life experience. And really what our internal models allow us to do is to anticipate and predict what's going to happen in the future. And so as a laboratory, we want to understand how the brain forms, builds, and uses internal models that are based off of recent life experience. And as I mentioned, we do this using the mouse as a model organism, and we do this by studying the sounds that the mouse's actions produce. So mice are naturally quite noisy creatures. They have a lot of natural sound generating movements. I'm showing one of them here, which is that mice produce vocalizations in many different social contexts, including courtship. And we study the natural and ethological sound generating behaviors that mice produce, including courtship vocalizations, but a lot of the work in our lab also studies a different type of sound generating behavior, which are engineered sound generating behaviors. So these are behaviors that we can have animals do in the lab, either because they will just do them naturally, like running freely on a spinning disc, or because we can motivate them to do it with a water reward. And these are behaviors that we've engineered to be extremely quiet. In fact, they don't make any sound at all, but we can monitor these behaviors in real time, and we can impose upon them any arbitrary acoustic consequences that we'd like. We tend to call these behaviors acoustic augmented reality. And what this allows us to do is to study how the brain learns a brand new sound generating behavior from scratch. And we can monitor the brain throughout an animal's entire life experience with this sound generating behavior, and it really simplifies the problem for us at an experimental side. So today what I'm going to talk to you about is one of these behaviors, which is shown at the far right. It's a mouse pushing a sound generating lever. And most of the experiments that I'm going to show you today were performed by a really exceptional postdoc in the lab named Nick Audette. And he had some help with this from a graduate student named Wenji Zhu, and I'll show you some photos of these folks at the very end. Cool. So what I first want to do is show you a video of what this behavior looks like. So you're seeing two cameras capturing the mouse simultaneously. The top one is really just getting a picture of the mouse's face, and the bottom one's showing you a bottom-up view of the mouse who's holding onto a little screw, a stationary screw with its left hand, and its right hand is positioned on top of a lever that it can move out. You'll see a big thing in front of the mouse's face. It's kind of a horseshoe shape. This is what we call a lickometer. It's an infrared beam that's monitoring whether the mouse is licking or not. And you'll see the mouse lick from time to time at this little black spout that's pointed at his mouth, which is a lick tube where he's going to receive a water reward whenever he successfully executes this simple lever behavior. And what you'll see from this video is that every time the mouse pushes the behavior, he hears a stereotyped self-generated sound. The sound is actually experimentally produced by us, but the animal feels ownership over the sound, which I'll show you some evidence for shortly, and believes it to be self-generated. Great, so I hope you can realize from that one of the really nice things about this is we can choose any sound that we want to superimpose upon this simple lever action. And in this video, it was the sound of a champagne cork popping. But when we do this in reality in the lab, we typically do this with a pure tone, a simple beep that the animal produces, and we train the animal extensively on that single beep. So here's a little diagram of what this looks like. So we've got this drawing of the mouse over on the left who's pushing this lever in order to receive a small liquid reward. But what the animal's movements actually look like is depicted over on the right. So what you're looking at here is the lever position on the y-axis over time on the x-axis. The animal starts with a lever near its body, what we call a home position. The animal's job is simply to push the lever out past a reward threshold. Once he passes that, he brings it back home toward the body. And when he gets back home, he gets a small liquid reward, okay? So the actual goal of this animal has nothing to do with sounds. It has nothing to do with processing sounds. The sounds just happen to be secondary to this behavior. So every time that the animal pushes the lever and he hears a sound at the reward threshold, and he pushes it to the reward threshold, he hears this self-generated sound that's predictable. It has a predictable sound frequency, therefore the acoustics are predictable and has predictable timing, right? So a lot like near you shutting the car door is a simple for-them behavior that has a predictable acoustic consequence that's stereotyped across renditions of the behavior. So what we do with this is we give the animal a few days of experience, a couple hours a day, producing this simple self-generated sound. So we'll give them 10 days, a couple hours a day. And what we want to know at the end of those couple of days is, do mice learn to expect the predictable acoustic consequences of their movements? So this first question is going to be a purely behavioral one. And so in order to test this, what we're going to do is after these 10 days of training on days approximately 11 and 13 is we're going to introduce deviant trials, right? So up until now, every time the mouse pushes the lever here is the exact same thing. But on days 11 and days 13, what we're going to do is on some subset of the trials, the mouse is going to hear something unexpected. So on one day, for example, we might have a small fraction of the trials be omissions. That is, the animal pushes the lever and instead of hearing a tone, he hears nothing. Or on another day, what we might have is frequency deviant trials. That is, the mouse pushes the lever and expects to hear a beep, but instead he hears a sound that's a boop. And the way we choose the boop is we shift the sound one octave away, either one octave above or one octave below the expected sound that the mouse expects the movement to produce. So we can monitor the mouse's behavior of the lever as well as his licking during these deviant trials. And we can make a couple of fairly formal predictions here. We can say that if mice have formed an expectation about the self-generated tone, then their behavior on deviant trials should reflect expectation violations. In the same way that if you and I shut a car door and we hear the wrong sound, we adjust our behavior, right? Now, prediction 2 is if the mice are using the self-generated tone to guide their behavior, that is, if the tone is behaviorally relevant to the animal, they might go above and beyond actually reacting to a deviant. And their reactions to the deviants might be directed and consistent across animals in the same way that you and I might be using the sound of a car door to adjust our behavior in a very stereotyped way. If we all push that car door and hear the clanking of a seatbelt in the door jam, we're probably all going to change our behavior in the same way, which is going back to the car, adjusting the seatbelt, and shutting the car again. Now, the mouse's behavioral changes aren't going to be quite that elaborate, but we should see some stereotyping nonetheless. Okay, so here's what this experiment looks like. Is at the beginning of the day, the mouse is going to push the lever and hear sounds that are expected. Then partway through the day, we're going to start to introduce omissions on a subset of trials. And we're going to monitor the mouse's behavior. In this case, what I'm doing is I'm distilling the mouse's lever behavior down to a single parameter, which is the length of the reach, right? So the mouse has learned to reach to a very particular length. He knows that's where he's going to get the reward, and then he's going to come back. And I'm focusing here specifically on omission trials instead of frequency deviant trials. But I can tell you that we can do the same analyses on frequency deviant trials and we see the exact same behavior. Okay, so for the first 20 movements that I'm showing you here, these are the last 20 movements before we started to introduce deviance, omissions. And then what I'm going to show you is the reach length on the next set of trials after omissions have been introduced. And what you can see is that if we look at interleave movements, that is movements that still have the sound, which are shown in the light blue or green, versus interleaved omission movements, we can see that when there's an omission, the mouse tends to push the lever farther and perseverate out there longer than when he hears the appropriate sound. And when he hears the sound, his movements are no different than on earlier trials before the omissions had been introduced. And we can quantify this for the mice. Here I'm showing you average lever trajectories during sounded trials, which is in the teal color, and omission trials, which is in the red color. And at the bottom, for 7 different mice, what I'm showing you is the reach length or the reach distance on the y-axis. That is, how much extra did they reach relative to normal trials on omission trials, as well as their movement duration. That is, how long did their movements last? And you can see that on average, mice tend to perseverate and reach longer when they hear an omission. We can also look at licking data. It turns out licking is also a potent indicator of expectation. We find that when mice have an omission trial, that they lick less and they lick slower, okay, right? So indicates to us that mice not only hear the sound, but they have an expectation for what that sound is going to bring later on in the trial. Okay, so with this tone heard at the reward threshold, as I introduce this task, mice recognize frequency, timing, and omission deviance. Their reactions to deviance are consistent across subjects. And we can tell that because the mice have this very stereotyped behavioral pattern across animals, that they're really using the tone to guide their behavior, right? When there's an omission, they stay out there and they kind of search for that tone that they're expecting. They use the tone in order to guide their reward searching behavior. Now, one of the nice things about this behavioral paradigm is we don't have to put the tone right at the apex of the movement like we've done it here. In fact, because it's a virtual paradigm, we can put that tone anywhere we want in the movement. We can train mice with a tone at different times during their lever trajectory. Mice will still make the same lever movement because that's what they need to do to get a reward, but the tone might happen earlier or later in the movement. And so here's one version where we can still have a highly predictable tone, but we can shift it very early in the movement, right after the mouse leaves its home position. And now what we've done here is the mouse is still going to have a high coupling between action and sound, but there's a much weaker coupling between sound and reward, okay? And what we find really interesting is that if we train a different subset of mice on this tone that happens earlier in their movement, and then we perform the same deviant trials, what we find is that mice still recognize deviance, but their reactions aren't guided in any way, shape, or form, right? They have all of the mouse's reactions are idiosyncratic. And so what that tells us from this is that mice do not use the tone to guide their ongoing behavior when the tone is earlier in their movement. And what's really important to us now is this gives us a very simple knob that we can turn, which is the timing of an expected sound. And that gives us the ability to change what we are going to functionally define here as the behavioral relevance of the self-generated tone to the animal. And I'm not going to go any deeper into this behavioral relevance, but we have a really cool set of data to show that the brain processes self-generated sounds almost like exactly the same, regardless of whether the sound is behaviorally relevant on the one hand, or if the sound is completely behaviorally irrelevant on the other hand, which really adds a potent piece of data to suggest to us that what the brain is really processing when it processes self-generated sounds is purely the expected consequences, as opposed to whether that sound is behaviorally meaningful or reward associated. Cool. Okay, so I've shown you that mice learn to expect the predictable acoustic consequences of their movements. What we want to ask next is, are predictable self-generated sounds processed differently by the auditory system than our other sounds? Okay. And so for this, I'm going to focus on this other deviant trial that we examined, which is frequency deviance, right? So on this day, what's going to happen is mice are going to begin the day by experiencing what they lever press movements that have the expected consequences, but partway through the day, we're going to introduce frequency deviance. And here it's going to be a little bit more of a dramatic perturbation. As soon as we flip the switch to introduce deviance, the mouse is going to hear 50-50. Some of them are going to be expected, some of them are going to be deviance, but they're going to be with equal probability. And that's an important statistical control that we do because it allows us to measure neuronal responses to expected and deviant acoustic consequences that are interleaved with one another. And any difference that we see in neuronal activity to those two sounds during the exact same behaviors cannot be accounted for by local acoustical statistics, things such as stimulus-specific adaptation, for example. Okay, so what we're going to do is, as the mouse is experiencing these frequency deviant trials, we're going to implant a multi-electrode array into the mouse's auditory cortex in order to do dense neuronal electrophysiological recordings. And what I'm going to show you here is neuronal responses to these two different sounds, both the lever-associated sound, which I'm going to call the expected sound, and the probe sound, which is this unexpected sound that the lever is going to produce. And we can look at neural responses to these sounds as the mouse produces them with the lever, but what I'm showing you here is even more simple. And it's responses to those same two sounds heard passively at the end of the day. So after the mouse pushes the lever a bunch of times, we're going to take the lever away and just play these two sounds back to the animal to measure tuning curves for individual neurons, and really to just gauge how strongly does the auditory cortex, or what's the capacity of the auditory cortex to respond to these two sounds when they're heard purely passively. And what I hope you take from this is that the auditory cortex of these animals that have been trained on a lever producing, on a tone producing lever, responds equally strongly to each of these two sounds in the passive condition, okay? But we see something different when the mouse hears those same two sounds when they're self-generated. And we see that in both conditions, the auditory cortex responds weaker to the sounds when they're self-generated compared to when they're heard passively. But there's this extra amount of suppression when the mouse hears the expected sound, right? And this jives really nicely with previously published work, both from our own lab on mice producing a very different behavioral paradigm running on a treadmill, as well as some really beautiful work from Torfi Sigurdson's lab looking at another behavior in which a mouse is pushing a button and hearing self-generated sounds, is that there's a preferential suppression of neuronal responses to expected self-generated sounds in the mouse auditory cortex. Now I'm showing you this at a single mouse level. We see this too if we aggregate across many different mice. And we can start to look at this at the level of individual neurons. I'm going to kind of take a deeper dive as we go into this talk at the individual neuron level. And we can measure at the single neuron level what's called a modulation index. And that is really effectively how differently does a neuron respond during the active condition versus the passive condition. Okay, we can calculate a modulation index. A modulation index ranges from +2 to -2. A value of +2 means that I should say a positive value means that a neuron is enhanced during movement compared to rest. And as you could probably guess from these population PSTHs that I showed you, that's going to be the minority of neurons that have that enhancement. And most of the neurons are going to have negative modulation indices. These are neurons that have suppressed neuronal responses to the same sound when it's self-generated compared to when it's heard passively. Okay. So this is what the modulation index values look like across a population of many hundreds of neurons, and you can see that there's a significant negative shift in these cells, that there's the vast majority of these cells are suppressed during movement relative to rest. Now this is only for the expected self-generated sound, so now we can look at the same distribution for neurons in response to the mouse making a movement and hearing an unexpected sound. And what we see is there's still a downward shift, but now there's significantly less suppression to the probe sound, that is the unexpected sound, than to the expected sound, okay? So again, this really jives with previous data in the literature showing that the self-generated sounds or expected self-generated sounds are strongly suppressed in the mouse auditory cortex. Okay, so I've shown you that at a population level, predictable self-generated sounds are preferentially suppressed in the auditory cortex. And what I want to finish with here is to start to look at how individual auditory cortex neurons process self-generated sounds. And one of the ways that we're going to start to do this is by taking advantage of the fact that the electrode arrays that we implanted into the auditory cortex were implanted perpendicular to the cortical surface, which allows us to penetrate through the auditory cortex and record from cells in each of the primary layers of the auditory cortex and delineate which neurons we were recorded from had cell bodies residing in different layers, in layer 2, 3, layer 4, layer 5, or layer 6. And this is interesting to us because we know that from a canonical cortical microcircuit point of view, different layers are thought to be involved in different aspects of cortical computations. And we also know that different layers of the cortex tend to receive different top-down signals, okay, right? So motor-related inputs tend to be concentrated outside of layer 4. They course into deep layers, layer 6 in particular, as well as layer 1, and they are also terminals in layer 2, 3, and layer 5, right? So we might think that neurons that reside in different layers of the auditory cortex might have differential responses to the intersection of movement and sound that the mouse is getting as they're producing this sound-generating lever behavior. So one thing we can do is we can look at how does modulation index, this value that we computed at the single cell level that tells us how suppressed a neuron is when they're processing one sound or another, how is that distributed across layers of the cortex? And so here what I'm looking at is the modulation index for the expected self-generated sound. That is, how much suppression is there to this expected self-generated sound compared to hearing the same sound passively? And we can see that as we go from more superficial layers to deeper layers, that there tends to be less and less suppression. This is really concentrated in layer 2, 3, and this really jives with previous work in the visual cortex in a similar but slightly different paradigm, looking at self-generated visual sensations. But to me, what's really striking is if I overlay on this graph now the modulation index as a function of layer for unexpected sounds. And you can see that some of the general trends are there, but there's a little bit more waviness here on this unexpected curve. And what I really want to draw your eye to is when the expected and unexpected curves deviate from one another, okay? So we can think of this difference is the difference in how much suppression is there to an expected sound versus an unexpected sound as a measure of the neuron's encoding of expectation violations or prediction errors. And so if I take each of these bars that I drew between these two lines, and if I simply plot them over on the right, we can see that these prediction error signals are really concentrated in layers 2, 3, and layer 5 of the mouse auditory cortex. And it turns out that there's a lot of cells that signal prediction errors. And what I want to do next is to walk through with you what some kind of the common phenotypes are that we observe in the mouse auditory cortex at the single cell level. Okay. So here what I'm showing you are a couple of example neurons on the left. There's a neuron on the top here and a neuron on the bottom, and each one is responding to the expected sound on the left and an unexpected sound on the right. And there's a total of 4 curves because each sound can be heard in the active condition, which is the light curves, as well as in a passive condition when the mouse is just chilling out, which is in the dark curves, okay? So this top curve is a very canonical auditory cortical neuron. This neuron has a fairly broad tuning curve. It responds to both of these sounds heard passively, which you can see from the dark curves. And it maintains a response to each of these sounds during the active condition, which is the light curves, but there's suppression to both of them. But that suppression is stronger for the expected sound, okay? This is a single neuron example that really matches the population phenotype that I told you about already. There's some other interesting example cells that we see as well. This is an example neuron that does not respond significantly, at least, to either of the sounds heard in the passive condition. Those are the 2 dark curves. But when the mouse produces a self-generated sound in either of these ones, these neurons all of a sudden respond to the sound during that behavioral condition, right? So these are auditory responses that only emerge when the mouse is actively engaged in the behavior. It's a really cool response. There's another one that I want to show you over here. This is a neuron that is unresponsive to the expected self-generated sound in either the active or the passive condition, okay? But it responds to the unexpected sound in both conditions. Okay, now on face value, this might seem like a very simple neuron. This is a neuron that is frequency tuned to one frequency over the other. In this case, the orange frequency is the frequency it responds to, and it doesn't care whether the mouse is moving or not. It just responds to that sound. And I agree with you that that is a true statement that we can make about this neuron. But what we find is really interesting is that we find significantly more of these neurons that respond to a sound agnostic of the condition in which it's heard for the probe sound, that is the unexpected sound, than we do for the expected sound. And to distill that finding down for you a little bit more, what that really means is that neurons that are responsive to the probe sound do not get suppressed when that sound is heard in the active condition. They retain their response. Whereas neurons that are responsive to the expected sound and the expected sound alone tend to be completely suppressed so that there's no longer a significant response when that sound is heard in the active condition. And that really exemplifies what we see at the population level that there's this massive suppression of the expected sound, but not of the unexpected sound, okay? Now, this last cell type that I want to show you is one that I think is doubly cool. And these are what we call prediction error neurons. So here's a neuron that does not respond to either of the sounds in the passive condition. Those are the dark curves. There's no statistically significant response. These would be what we might refer to as nominally non-responsive neurons, which have been characterized by my colleague here at NYU, Rob Franke. What's really cool too is if the mouse pushes the lever and hears the sound that it expects to produce, we also don't get a response from these particular neurons. But if the mouse pushes the lever and hears an unexpected sound, you get a reliable response. And so these neurons only fire in one condition. That is when the mouse is pushing the lever and hears an unexpected sound. So we would call these prediction error neurons. And what's really cool is that we focus over here, we see significantly more, something like 5 times as many neurons that respond to prediction errors. That is, they respond to an unexpected self-generated sound. Then we see neurons that respond only to the expected sound. And we see that these neurons tend to be really concentrated in layer 2, 3, and layer 5. And that really leads us to thinking that these 2 layers of the neocortex might be particularly important for integrating motor and sensory informations in order to make and facilitate expectations about what sounds are expected to be heard given our movements. Great, so I want to finish by quoting Yogi Berra here and say that it's tough to make predictions, especially about the future. And completely agree with that, but I think we've got a really cool paradigm here in which we can start to understand how the brain learns to form expectations and make predictions about the future. And so what I've shown you so far is that mice learn to expect the simple acoustic consequences of a simple behavior, that the auditory cortex has neurons that are sensitive to expectation violations. I showed you neurons that are sensitive to frequency violations, but we also have neurons sensitive to timing and movement violations. And that prediction error signals tend to be concentrated in specific auditory cortical populations, namely in this case, layer 2, 3, and layer 5. There's still a lot of cool things that we're trying to answer, such as what is the source of the motor-related input that allows cortical predictions to arise? Which neurons and which synapses are changing over time as mice learn the sound of a movement in order to store that internal model that relates an action to its outcome? And are the same predictive circuits that we're studying in these simple behaviors also engaged during natural sound generating behaviors such as vocalizing? So I want to just finish by saying that it's hard to imagine a meaningful behavior that doesn't have expected consequences. And it's, I think with every behavior that we make, we're subconsciously comparing the outcomes of those behaviors with the expected outcomes of those behaviors. And so we think that we have a really cool system here for trying to tap into some of these very foundational neurobiological questions about how the brain operates as we navigate through our daily world. So let me finish by just thanking the members of the lab, in particular Nick Audette, a postdoc in the lab who really spearheaded.
Hi, everyone. My name is Daniel Reznik, and I will describe to you a series of studies that I did when I was still a PhD student in Tel Aviv University, Israel. And during that time, I tried to understand and characterize how human auditory perception is modulated by voluntary action execution. And when I tried to project myself back then, I think that the main inspiration that drove my scientific inquiry to that particular course was a book, Phenomenology of Perception, by a French philosopher, Maurice Merleau-Ponty, that I read during my military service in Israel. And in this book that was published in 1945, Merleau-Ponty presents a very nice idea that I found quite fascinating back then as a teenager, that our sensory perception is not a veridical reflection of the world around us, an idea that might sound very counterintuitive, but it is a rather product of dialectical interaction between intentional and internal state of the perceiving subject and the sensory objects that are being perceived. And since then, I was quite preoccupied by this fundamental question of how differently we perceive sensory stimuli that are the direct product of our actions and our intentions, namely active perception, compared with physically identical stimuli that are generated by someone else, by something else, that are lacking this internal self-generated drive, namely passive perception. So this notion of active versus passive perception has been studied already for many decades in humans and in animals using various methodologies and various modalities. And in humans, I think that one of the first demonstrations for this differential processing was a very nice EEG science paper by Schaffer and Marcus, published in 1973, in which they showed that the amplitude of the N1 evoked potential was reduced in response to auditory stimuli, to clicks that participants triggered by their own actions, compared to identical tones, clicks that were generated by a machine. And here on the right, you can see a graph from this paper showing the N1 amplitude by a machine trigger stimulation and the amplitude of N1 component for self-generated stimulation. Now, if we move about 4 years forward, so we can see that there's also enough behavioral evidence showing that also the perceptual level auditory processing is modulated by self-generated sound actions. Namely, a series of studies led by Carmen Weiss showed that the perceived intensity of self-generated sounds is reduced compared to identical sounds. So, namely, we perceived sounds that were generated by our own actions as to be softer compared with sounds that were generated by someone else, by either experimenter or a computer. But on the other hand, studies by Benjamin Morillion suggest that actually frequency discrimination is improved when sounds coincide with finger tapping, showing that motor actions can either attenuate or enhance auditory processing. In other modalities, there's this famous notion that we cannot tickle ourselves, but we can easily tickled by someone else. And on the neuroimaging level, there is evidence showing that self-generated tactile stimulation modulates fMRI responses in primary and secondary somatosensory regions, again, giving more support to the notion that sensory processing is modulated, whether it's a product of self-generated actions or just passively perceived. Now, even though this talk would be almost solely focused on human neuroimaging, I do want to highlight one particular animal study of, I think, really great importance, because in a very elegant and clear way, this study demonstrates a direct anatomical projections from motor to auditory cortices in mice. So here in left, you can see injection site of retrograde labeling in auditory cortex, and here to the right, you can see the labeled cells in mice M2. And even though obviously it's not so it's not a trivial task to draw direct parallels between mice brain and human brain, I think this finding is giving us an exciting anatomical framework within which the behavioral and neurophysiological modulations in humans that we discussed about might operate. So when I started my PhD, surprisingly, there were almost no works showing how auditory perception and auditory processing in humans affected by self-generated actions using whole brain imaging. So to that end, this was kind of low-hanging fruit that we want to address, and we designed our first fMRI study to address the question of what brain regions involved in sensory modulation during voluntary action execution, and how these brain regions interact between themselves. So to this end, we designed a very nice and a very fun experiment that our participants very enjoyed performance. So we had a very nice MRI compatible piano keyboard. So our participants could actually play short piano tunes inside the scanner. So we composed 2 short tunes for the right and for the left hand that the participants learned beforehand before entering the scanner. And then during the scanner, we just ask them by visual cues to play either right hand tune or left hand tune. And we also recorded the performance of every participant. And then after the active condition, we actually replayed to the participants their own performance with all the idiosyncrasies, with all the idiosyncratic performance and all the mishaps. And so at the end, the participants basically heard identical sensory input, but in one condition, it was generated by their own actions, and in the other condition, it was just passively heard. So since we were interested in auditory cortex, so we defined the auditory region of interest using auditory localizer. And then we looked at this region of interest, fMRI activity in active and passive conditions, and we saw that fMRI activity was enhanced in auditory cortex during active sound generation compared with passive listening. So you can see the average group data on the right in bar graph, and individual subject data on the right, broken into right and left hemispheres. We also controlled for the repetition effect. So we also had a condition that we just expose our participants to the same tune twice, just to see if just second exposure to the same musical sequence will lead to a reduced fMRI activity in auditory cortex, something that might explain this enhancement in the active condition compared to the passive condition, but we didn't see such evidence. And again, suggesting that this enhancement has something to do probably with the motor component and with the active sound generation. Now, if you remember, our participants they performed the musical tones with the right and left hands. So when we break the active condition into right and left hands tune, so we can see a very nice double dissociation between the hemispheres. And we can see that the left auditory cortex was enhanced during right hand performance compared with left hand performance, basically when the hand was contralateral to the auditory cortex, and vice versa for the right STG. For the right auditory cortex, you can see that the activity there was enhanced during left hand performance compared with right hand performance. So again, on the left, you can see the group average data, and on the right, you can see individual subjects, again, broken by right and left STGs. And you can see that the right STGs, right auditory cortex go with the left hand, and left auditory cortex goes with the right hand. Now, this sensitivity of auditory cortex enhancement to the identity of the executing effector was also further confirmed by function connectivity analysis that we did on the whole brain level using seeds at either right or left motor cortices, and to validate the GLM findings, we saw that the right motor cortex was most connected with right superior temporal gyrus, and the left motor cortex was connected the most with the left superior motor gyrus during right and left musical sequence execution. So this provided more evidence that the origin of the auditory modulation that we saw in the active condition originates in the motor cortex, but we were still not quite convinced, especially given evidence linking closely auditory perception and tactile stimulation. And as we can already understand, one inherent component of motor actions is somatosensory feedback, right? So I press a piano key, I press any key to trigger a button, there's always a somatosensory feedback from this act of pressing. And especially given evidence that the tactile stimulation, for example, affects auditory sensitivity, and that touch triggers fMRI activity in auditory cortex. And here we can see the GLM map from the study. it is still unclear that the origin for the auditory modulation that we saw previously actually originates from the motor component and not from the somatosensory component that is inherently accompanying the motor action. So we designed another study just to address this issue. And in this study, we used another pretty cool toy. So we used an MRI-compatible motion detection glove that we programmed in such a way that our participants could basically play musical tunes by just moving the fingers in the air without touching any key. And I really hope that you could hear this. Let's see. Yeah, and by this we aim to decouple the motor and somatosensory component and to see and to examine if we will still see the same modulation of auditory cortex during the glove condition compared with pressing a key condition. And the first thing that we did, we wanted to validate that we indeed have less somatosensory feedback during the glove condition, and we can see the resulting contrast here. So we can see this patch in left primary somatosensory cortex, indicating indeed that during a key condition there was more somatosensory feedback compared with the glove condition. And when we look at the activity, at the auditory cortex, so we basically replicated our findings from the previous study by showing enhanced activity in the active condition compared with the passive condition in the auditory cortex contralateral to the executing hand, namely in the left superior temporal gyrus. Right superior temporal gyrus didn't show any difference. But most importantly, that when we compare the magnitude modulation of active versus passive, so we can see that there was no difference between the modulation in key condition compared with the modulation in the glove condition. Namely, that even if we reduce the amount of somatosensory feedback, the modulation of auditory cortex remains the same. And I think this is compelling evidence showing that the origin for this modulation is motor indeed, and the contribution of somatosensory component is secondary. So after convincing ourselves that this is indeed mostly motor and not somatosensory, for example, we kind of switched our perspective. And we focused on the sensory meaning of the motor signals that are being conveyed from the motor cortex to the auditory cortex during execution of actions that are associated with auditory consequences. And then our next question was, so what does the activity in motor cortex during voluntary action execution actually represent? So here we came back to basic, to the classical studies from the very first days of this active versus passive studies, and we came back to electrophysiology and to the EEG and MEG, and to this end we designed two very simple studies, and in one of them we wanted to focus on readiness potential, namely on the preparatory activity preceding motor execution. And to those of you who are not familiar, so basically readiness potential is an accumulating negativity over motor regions occurring prior to action initiation, prior to action execution. Well, the original name for it is Bereitschaftspotential, and now when I live in Germany, I actually know how to pronounce it. So that's pretty nice. And therefore, we can basically divide this entire time course into pre-action activity and the preparatory readiness potential part, and the post-action evoked sensory component. And since we wanted to see whether preparatory motor activity already bears some sensory information about the upcoming sensory stimuli, we wanted to focus on this blue area. And therefore, we designed a simple study in which we ask our participants to press a button, to press two buttons with either the index finger or middle finger. And in one run, the button presses with the index finger were associated with a sound, and the button presses with the other finger were not associated with anything, so they were just silent. And in the other run, just the mapping was reversed. And in the final run, we just had participants listen to passively delivered sound at expected intervals, just to control for the effect of auditory expectancy. And since we wanted to look whether preparatory activity already contained some information about the upcoming sensory stimuli, we asked our participants to maintain a certain delay that we manipulated to be at least 3.5 seconds between each press. And then during the data analysis, we basically focused on this time interval preceding action execution. So we saw this, and I really like this. I really like this graph. So basically, we showed that the readiness potential preceding button presses that were associated with auditory consequences was more negative compared with readiness potential that was prior to button presses that were not associated with auditory consequences, basically with silent presses, and that this effect could not be explained by auditory expectancy. So this is the passive condition, the orange line, and we cannot see any early negativity. So basically, this finding indicates that motor preparatory activity already represents some information of the upcoming sensory stimulation, but it still lacks some information, right? Because we do get information of what happens before action execution, but what about what happens during action execution? And for this we delivered. And for this we designed another study, and this was an MEG design. And one inherent limitation for studying the motor signals alone without the reafferent activity is that typically when we perform a motor action, we immediately get sensory feedback, right? I press a piano key and I immediately hear feedback. I press a button, I press a key, I press a doorbell, and I immediately hear and the ring immediately rings. So what we did, we introduced a constant temporal delay between the action and the auditory consequences. So in this MEG design, auditory consequences were near threshold auditory near threshold tones. So because we did an auditory detection task, but by introducing this constant delay of 500 milliseconds, we could actually decouple the motor signal from the auditory reafferent signals. And in the active condition, the sounds were triggered by participants' voluntary actions, and in the passive condition, the cue indicated the onset of the 500 millisecond cue interval. Sorry. So what we saw is the following. So again, in the left auditory cortex, again, remember that the participants performed the actions with the right hand. We saw this very nice transient increase in auditory cortex following button press in the active condition. Importantly, this increase was not present in the passive condition. Again, something that could not be associated. So it means that this increase in active condition could not be associated with sensor expectancy. And more importantly, we see that the increase in auditory cortex activity during active condition is much lower than the induced activity during motor only condition, meaning that even during action execution, motor actions that are associated with sensory feedback also convey information about this upcoming feedback that goes beyond the purely motor component that is represented by this gray line. Yes, and in right auditory cortex, we didn't see any modulation. So just to summarize so far, and basically to summarize this entire talk. So I showed evidence using fMRI that modulation of sensory cortex during perception of self-generated stimuli originates in motor cortex, and it is sensitive to the identity of the executing hand. And using EEG and MEG, we've shown that activity in motor cortex prior and during execution of voluntary actions represents their sensory consequences. We also have plenty of behavioral findings, but I think we could present them and discuss them during the discussion and not here. And some future steps that I think might be interesting to explore. So first, there's another fundamental question of how expectations shape perception. Even though this is a bit similar question, but it's also different because we did not explicitly manipulate expectation levels in our paradigms. And for this, I strongly recommend you to read works by Daniel Yon. We also might consider this interesting transition from simple predictive models to complex predictive models, because all the data that I presented here are from very simple paradigms of making sensory predictions at the level of 500 milliseconds, or just making immediate prediction. And we can see how the brain solves this problem in using the motor auditory circuitry. But it's interesting because we can also build predictive models that are much more complex and much more abstract, right? Because where I will spend my next vacation, will Ukraine join NATO, or how certain people will respond in certain situations, and therefore it's interesting to try to extrapolate these findings from how motor sensory predictions modulate sensory processing and sensory perception to much more abstract and complex future models of future thinking and other cognitive domains. So I want to thank all these people who supported me during my PhD, especially Rimu Kamer, my PhD supervisor, and Elana Tsiongolombik. And of course, thank you for listening, and we all will be available here for questions. Thank you so much.
Hi, everyone. Thank you for listening. I am Ana Pinheiro from the University of Lisbon, and in this talk, I will discuss sensory feedback processing in auditory verbal hallucinations. Specifically today, I will present one of our EEG studies that examine how sound feedback is perceived in psychotic patients with auditory verbal hallucinations and in non-clinical voice hearers. Briefly, I will try to convince you that selective changes in sensory feedback to the voice are core to auditory verbal hallucinations, and that these changes already show in preparatory motor activity. Despite an increasing number of studies in the last decades, auditory verbal hallucinations remain one of the most intriguing phenomena. So why do some people hear voices in the absence of corresponding external stimulation? In other words, how does the brain create such a complex perceptual experience? A prominent hypothesis is that auditory verbal hallucinations are a consequence of altered prediction of the sensory outcomes of one's own actions, such as talking, which then affects the distinction between self-generated and externally generated stimulation. In the case of speech production, the auditory system relies on sensory feedback to distinguish between one's own and other voices. And more specifically, when an action is performed, an efference copy of the respective motor commands containing information about the expected sensory feedback is presumably sent to the sensory systems where a corollary discharge of the expected sensory consequences of the motor act is compared to the actual sensation. If they match, sensory responses are attenuated. Sensory suppression is therefore typically taken to reflect internal forward modeling. Studies that have tested psychotic patients with auditory verbal hallucinations reported reduced N1 suppression, that is a similar processing of self-initiated and externally produced sounds, which is shown in a lack of difference between the blue and red lines here in the grand average waveform. This indicates a failure to distinguish between internally and externally generated sensory signals, such as one's own voice. Neuroimaging studies have also supported a lack of differentiation of temporal lobe activity when listening to self-voices compared to other voices. Studies with the same patients using behavioral measures have consistently reported external misattribution errors. That is, an increased tendency to consider that pre-recorded self-voice stimuli were produced by somebody else, indicating altered self-other voice discrimination. Despite the contributions of all these studies, it remains to be clarified whether similar alterations are found in non-clinical individuals who report hearing voices by directly comparing psychotic and non-clinical voice hearers. This would allow us to test the psychosis continuum hypothesis, which postulates that psychotic symptoms exist on a continuum in a general population, and that auditory verbal hallucinations in psychotic and non-clinical voice hearers engage similar neural and cognitive mechanisms. We use the EEG to investigate this question. In this EEG study, we tested 16 non-clinical voice hearers, 16 psychotic patients reporting auditory verbal hallucinations, and 15 control participants. Non-clinical voice hearers were selected based on their scores on the Lowney-Slade hallucination scale, which is widely used, and after extensive clinical assessment. The clinical interview established that voices were distinct from thoughts, were unrelated to drug or alcohol abuse, and that participants did not have a psychiatric diagnosis in relation to voice hearing. All participants completed the button press task, in which we tested the effects of stimulus type. In other words, we examined whether the putative alterations in sensory suppression in auditory verbal hallucinations are specific of vocal compared to non-vocal sounds. The task included 3 conditions, as shown here. In the auditory motor condition, participants pressed a button that elicited a sound, and the sound was either a pure tone or the pre-recorded voice of the participant. In the auditory-only condition, these sounds were passively presented. The motor-only condition was a control condition in which participants pressed a button, but no sound was elicited by the button press. The motor activity of the motor-only condition was then subtracted from the auditory motor condition to control for motor artifacts related to the button press, which is a typical procedure in this type of paradigms. The study I am going to show you tested how hallucination proneness had an impact on event-related potential component modulations, informing about different operations of the forward model. The N1, indexing the comparison of predicted and actual feedback, the P2, reflecting the conscious detection of a self-initiated sound, and the readiness potential preceding the action, reflecting afferent copy formation. We predicted that hallucination proneness would be more strongly associated with alterations in the comparison of predicted and actual sensory feedback. And as for potential effects of auditory verbal hallucinations on the readiness potential and on the P2, our approach was exploratory. When comparing the event-related potential responses to tones, we observed no condition-dependent group differences. In other words, the N1 was suppressed in response to self-triggered tones compared to externally presented tones in the three groups, which we clearly see in these when average waveforms. The dotted line indicating the ERP waveform to self-generated tones shows a less negative N1 than the solid line around 100 milliseconds post-tone onset. The N1 was nonetheless generally reduced in patients in line with prior studies. When comparing the event-related potential responses to the self-voice, we observed important group differences that were enhanced during the processing of self-triggered voices. We observed the expected sensory suppression effect in controls as for tones. However, in non-clinical voice hearers, the N1 pattern was reversed. That is, the N1 was enhanced in response to self-triggered voices compared to externally presented voices, which is indicated by a more negative voltage reached by the dotted line than by the solid line. Confirming previous studies, the N1 suppression was reduced in patients. Regarding the P2 component, we observed the same pattern in the three groups. That is, the P2 was suppressed in response to self-triggered compared to externally presented voices, as shown here. Nonetheless, there was a larger P2 suppression in non-clinical voice hearers, shown here by a larger difference between the auditory-only condition or externally presented voices and the auditory motor condition or self-triggered voices. We also found that the N1 suppression effect was predicted by the readiness potential. In other words, the larger the readiness potential, the larger the N1 suppression. Specifically, we observed that motor activity preceding the button press was reduced in non-clinical voice hearers as well as in psychotic patients with auditory verbal hallucinations compared to controls. And we observed that motor activity preceding the button press was reduced in non-clinical voice hearers, specifically for sensory feedback to the self-voice, which was reflected in an effect of stimulus type. So together, these findings indicate that altered voice feedback is core to auditory verbal hallucinations. We observed that alterations of sensory feedback processing in both psychotic and non-clinical voice hearers are related to changes in preparatory motor activity, providing partial support for dimensional models of psychosis. These alterations are reflected in the readiness potential, the N1, and the P2. Importantly, these findings additionally suggest that specific alterations in the processing of self-generated vocalizations may establish a core feature of the psychosis continuum. A voice-specific rather than generalized altered sensory prediction may explain why voices represent the most common abnormal perceptual experience in subjects who report auditory verbal hallucinations. As a consequence, sensations that should have been predicted but were not might take on inappropriate salience, providing the basis for an auditory verbal hallucination. Another critical question is how can we explain these alterations from a circuit perspective? In particular, the cerebellum is thought to play a key role in the comparison of predicted and perceived sensory feedback and update of the forward model. However, the role of the cerebellum in auditory verbal hallucinations has remained largely peripheral in the literature so far, which is potentially due to a corticocentric bias, as seen in optimized cortical scanning and analysis protocols that often neglect the cerebellum. Its putative contributions to auditory verbal hallucinations were discussed in a review paper that we have published last year. Consistent with the role of cerebellar dysfunction in auditory verbal hallucinations, evidence from symptom capture studies shows that hallucinated voices are associated with increased cerebellar activation. Therefore, to evaluate the robustness and regional specificity of cerebellar changes as a function of auditory verbal hallucinations, we recently conducted an activation likelihood estimation meta-analysis of structural and functional connectivity studies using ginger ale software. The combined studies included 1,360 patients with psychosis and 794 controls. The results reveal 3 clusters showing convergence of neuroimaging findings. The first centered in left CRUS-1, the second centered in right level 5 and 6, and the third centered in right CRUS-1. So these findings suggest that cerebellar disconnectivity could indicate a specific liability for auditory verbal hallucinations, particularly in sensory motor and cognitive cerebellar zones. These abnormalities may contribute to altered sensory feedback processing and consequently affect higher level cognitive functions, for example, cognitive control in auditory verbal hallucinations. So the findings discussed in this talk support the contribution of neural mechanistic changes in relation to the forward model in auditory verbal hallucinations, which seem to be at least partly associated with cerebellar dysfunction. They also support the relevance of investigating sensory motor alterations in psychopathology. We are currently extending our EEG studies with complementary neuroimaging techniques and computational modeling. So stay tuned. Thank you very much for your attention, and thanks to a long list of collaborators in Lisbon and in Maastricht. I look forward to hearing your questions.
Hello, my name is Silvia San Miguel, and I am an associate professor at the Brain Lab at the University of Barcelona. Today, I will talk about the impact of our actions on auditory processing and memory encoding of sounds. All our actions have sensory consequences that we can refer to as self-generated stimuli. Sensory feedback in the form of self-generated stimuli allows us to evaluate the success of our actions towards achieving the goals of the actions. However, not all self-generated stimuli are intentional. Most actions also have sensory consequences that are secondary to the main objective of the action, like, for example, the sounds that we make while washing the dishes or the sound of our own footsteps. In any case, we humans are able to relate the consequences of our actions to ourselves as the causing agents of those consequences. But the exact specific mechanisms that underlie this important function are yet not completely clear. So my main line of research over the last years has been studying self-generated stimuli and specifically self-generated sounds. And until recently, I have focused on studying the sensory processing of self-generated sounds, as the other speakers of this symposium have been talking about. However, the ability to differentiate self-generated stimuli and to give them a special processing can have important consequences at other levels. And specifically, it makes sense to assume that if self-generated stimuli are processed in a differential manner, then probably they will also be encoded in memory in a different way. So in this talk, I will first make a very brief overview of the state of the art regarding the sensory processing of self-generated stimuli, especially focusing on sound. And then I will present results from 3 experiments in which we try to elucidate the effects of self-generation on memory. So what do we know about the sensory processing of self-generated stimuli and of sounds? There's a great deal of findings that have shown that our sensory systems respond different to stimuli when they are generated by ourselves. Often the fact that it is difficult to tickle ourselves, although we feel ticklish if someone else is producing the same sensory stimulation, is used as an example to illustrate this idea. And indeed, the most established finding is that sensory responses to self-generated stimuli are attenuated. And this is also the case with self-generated sounds, with many studies showing that the auditory event-related potentials are suppressed when sounds are self-generated. Here we can see an example. In the dotted line, you see the self-generated sound, and in the solid line, you see the externally generated sound, and you see the self-generation effect, which is the attenuation of the auditory N1 component. So this is commonly referred to as the self-generation effect, and it's generally explained from a predictive coding perspective, given that self-generated sounds can be better predicted than externally generated sounds. So the most accepted explanation to what underlies these self-generation effects is that whenever we execute a motor command, together with a motor command, we also generate always a prediction of the expected sensory consequences of executing that command. So if the command is to clap my hands, I should expect to hear the clapping sound. Now, when the motor command is executed and the stimulation arrives, the sensory input is then compared to the predicted input. And responses to any sensory input that matches the predicted input are suppressed or canceled out. This explanation fits very well with predictive coding models of brain function, according to which the afferent information flow in the sensory hierarchies contains only the prediction errors. So these models are proposing that there are sensory responses only to the part of the sensory input that cannot be predicted, or in other words, the prediction error. However, we also know that during movement, many physiological parameters are modulated. And this includes modulations of sensory responses. And I refer to these as unspecific effects because they don't depend on a specific association between a particular movement that causes a particular stimulus. But rather, in this case, movement can affect all sensory processing in a general way. And this has been mostly shown in animal studies, comparing responses during locomotion as opposed to during rest, but also during other types of movements. Now, in humans, self-generation effects on sound processing have mostly been studied comparing responses between sounds recorded in a block in which participants repeatedly press a button, and with every button pressed, a sound is generated, and comparing this to responses to sounds recorded in a separate block in which the participant just listens passively. So in this situation, there is a strong action effect association, since the sounds are contingent on the actions. And here we can assume that self-generation effects are due to prediction, and this is where we observe indeed the typical suppression of the auditory event-related potentials. However, by now there are many studies that have also shown that self-generation effects are also found when there is no such contingency between the actions in the sounds. And in these studies, participants make a series of button presses, and unrelated to the button presses, a series of sounds is presented. And in this situation, some of the sounds will temporarily coincide with the action, but are not generated by the participant's action. And since there is no contingent relation between the action and the sound, they can also not be predicted from the action. But nevertheless, the sensory responses to the sounds that randomly coincide with the execution of the action are also suppressed. Although we have some indication that the contingency effects and the coincidence effects show some different patterns of modulations, as we can see here. So our idea is that the self-generation effect is probably rather a compound of different effects that perhaps affect different types of self-generated sounds differently. So surely when the main goal of our actions is to produce the sounds, and thus we have a very strong internal model of the action effect associations, prediction is probably playing a very important role in the modulation of the sensory responses. And according to predictive coding models, prediction will mainly act by canceling out the responses to the predicted stimuli. On the other hand, a possible candidate mechanism for unspecific effects of movement on sensory responses is the activation of subcortical neuromodulation mechanisms. And in particular, we hypothesize that the locus coeruleus norepinephrine system is activated by movement, causing an unspecific gating of sensory responses. And perhaps this type of modulation is more relevant when the self-generated stimuli are just an unintentional byproduct of our actions, like in the example of the sounds that we make while washing the dishes, or the sounds made by our footsteps. Now, in parallel to the research on the sensory processing of self-generated sounds, other researchers have investigated memory processing of sounds. And using very different experimental designs to those employed to show the self-generation effect on sensory processing, memory studies have shown that self-generated stimuli are remembered better. And this effect is referred to as the production effect. And it has been mainly shown in two types of paradigms. In studies using word lists, participants have to learn a list of words, and some of these words are read silently, while some of these words are read aloud during encoding. And it turns out that words that are read aloud show up to 10% benefit on a later recall test. Other studies have shown that when participants have to learn a melody, they remember it better if they can play it themselves on an instrument compared to if they listen passively to it. And then they are better able to detect changes to the melody if they were able to play it on an instrument. So a previous study suggests that self-generating the stimuli helps to encode them in memory and confers a memory advantage. But other findings might suggest the opposite. So as we saw before, one of the main explanations for the self-generation effects on sensory processing is that self-generated sounds are more predictable, since they can be predicted quite precisely based on the motor commands. But in the memory field, there are many findings that show that unpredicted stimuli are remembered better than predicted ones. So from this perspective, it would be expected that self-generated stimuli, which are more predictable, would be remembered worse, not better, like the production effect studies show. So altogether, we have seen that actions have an impact on sensory processing and on memory, but there are many open questions regarding the underlying mechanisms, and it has not been explored whether and how the self-generation effect on sensory processing might relate to the production effect on memory. So the main aim of our research line at the moment is to try to link these two phenomena, asking the question, are the production effects on memory of enhanced recall for spoken words and played melodies a consequence of the altered sensory processing that these stimuli have received? That is, the suppression of the sensory responses, both when the sounds are contingent on the actions and when there is no contingency and they only temporarily coincide with the action. And we are particularly interested in trying to unravel the physiological mechanisms behind these effects and understanding the possible contributions of predictive mechanisms and of unspecific neuromodulation mechanisms. And to do so, we use auditory memory tasks in which some of the sounds to be remembered are produced by the participant or coincide temporally with an action of the participant. And some of the sounds are externally generated and do not coincide with an action. We measure memory performance for these sounds and sensory responses using event-related potentials, and also pupil dilation as a proxy measure of the activation of the locus coeruleus norepinephrine system. So I will now present 3 experiments in which we have used this approach. In the first 2 experiments, we have investigated the effects of action-sound coincidence on sensory processing and memory encoding. So in these experiments, the sounds to be remembered may coincide with an action, but the sounds cannot be predicted based on the actions performed. In the first experiment, we use a delay short-term memory task. Every trial starts with an encoding phase in which 6 sounds will be presented, and half of them will coincide with an action, and we refer to this as motor auditory, and half will not coincide with an action, and we refer to this as auditory only. So to achieve this, participants are presented at the start of the trial with several rectangles positioned in a row at semi-random distances from each other. When the trial starts, a line is going to start advancing from left to right. And every time this line crosses one of the rectangles, participants have to press a button, in total 6 times. Now, half of these button presses are going to be accompanied by a sound, so the motor auditory events here. while half of them are not going to be accompanied by a sound, and these are the motor-only events. And another additional 3 sounds are presented in between the button presses, and these are the auditory-only events. So altogether, participants hear 6 sounds. Half of them are motor auditory, and half of them are auditory only. And they must maintain in memory all these 6 sounds during a short retention period. Then at retrieval, a test sound is presented that was part of the encoding set on 50% of the trials. And participants have to respond whether the test sound was presented during the encoding phase of this trial. So with this design, we can compare sensory responses of sounds that coincide and don't coincide with an action at encoding to assess the self-generation effects. And then we can measure at retrieval whether the motor auditory sounds are remembered better or worse than the auditory-only sounds, and whether this depends on the sensory processing at encoding. So what did we find? At encoding, we observed the typical self-generation effects with attenuated N1 and P2 responses for sounds coincided with an action in red compared to those not coinciding with an action in blue. Here in gray, you can see the difference between the two, so the self-generation effects. However, despite we found these very strong self-generation effects at encoding, this differential sensory processing did not appear to have any consequences for the memory encoding of these sounds. So we found no differences in memory performance between sounds that coincided with an action during encoding and sounds that did not coincide with an action during encoding. So we were not able to find a production effect with this design, which is quite removed from the task in which this production effect has been described. But this also seems to suggest that there is no relationship between the self-generation effects on sensory processing and the production effects on memory. However, to be sure, we decided to try again in a second experiment with some minor. So in the second experiment, the encoding and retention periods were the same, and we only modified the retrieval phase. Here, we presented 2 instead of 1 test sound, and participants had to make a 2 alternative forced choice between the 2 and to decide which one had been presented at encoding. And unbeknownst to the participants, in fact, in most of the trials, both test sounds have been presented at encoding. One coinciding with an action, the motor auditory one, and one not coinciding with an action, the auditory only one. So these trials that we refer to as the 2T for 2 test sounds at encoding trials allowed us to measure whether there was a memory bias towards one or the other, and also to be able to compare ERP responses between sounds that were encoded as auditory only and sounds that were encoded as motor auditory, but when they are presented passively as test sounds at retrieval. In any case, we also did include some trials in which indeed only one of the 2 test sounds was presented as encoding, while the other test sound was a new sound. And so these trials allowed us to have a veridical measure of recall. At encoding, we replicated the N1 and P2 attenuation for sounds coinciding with a button press. And here, we also measured pupil dilation. And as we hypothesized, we found greater pupil dilation when participants pressed the button. Thus, these actions were likely accompanied by a burst of noradrenergic neuromodulation reaching the cortex that we assume could be related to the sensory suppression effects. At retrieval, when both of the test sounds had been presented at encoding, we found no memory bias towards one or the other. So sounds that coincided with an action during encoding and those that did not coincide with an action during encoding were just chosen randomly, each on 50% of the trials. However, when only one of the 2 test sounds had been presented at encoding and the second was a new sound, we did find a better memory performance for sounds that did not coincide with an action when they were presented at encoding. So the opposite of the production effect. One possible explanation for this effect is that at retrieval, all the sounds are presented passively. So it is known that test stimuli that are presented out of context, out of the context in which they were encoded in memory tasks, are remembered worse. So this could explain here worse memory for sounds that were encoded with an action, but are later presented without the action at retrieval. In this experiment, we also measured ERPs elicited by the test sounds at retrieval, and we compared sounds that had been encoded as motor auditory, coinciding with an action, to sounds that had been encoded as auditory only, so not coinciding with an action. And what we found was enhanced sensory responses for sounds that had been encoded as motor auditory, and in particular, an enhanced TB, which is a temporal subcomponent of the auditory and one wave. This modulation, however, was only present for sounds that were remembered and not for those that were forgotten. So if we interpret enhanced sensory responses as increased prediction error following the predictive coding models, this finding might be consistent with this interpretation that stimuli that were encoded with an action are presented out of context and retrieval. And so what we see is that they elicit an increased prediction error response. So from the first two experiments, we have learned that production effects are not present, or that might even be reversed in the setting of our task when there is no contingent relationship between the actions and the sound. Despite we do find clear self-generation effects on sensory processing, and although depending on how it is measured, actions do seem to have an impact on a later memory recall. So to try to understand this relationship better, in the third experiment, we introduced contingency between the actions and the sounds, so making it more similar to the setting in which production effects have been reported. Now, in most previous self-generation and production effect studies, the self-generated sounds both coincide with an action, and they are also predictable based on the action, while the externally generated sounds do not coincide with an action, and they are also unpredictable. So in this experiment, we wanted to try to dissociate and compare the effects of coinciding with an action from the effects of predictability. And thus, we manipulated these two variables in an orthogonal 2x2 design, and then we measure effects on sensory responses and on memory performance. So to achieve that, participants performed a short-term memory task in two separate sessions, a predictable session and an unpredictable session. Now, as in the previous experiments, each trial started with an encoding phase in which sounds could be externally or self-generated. In the predictable session, there were two types of events during encoding, self-generated sounds and externally generated sounds. On the screen, participants could see a fixation cross and 4 unfilled circles of different colors that corresponded to the 4 response buttons that they could use on a response pad. Each event started with a fixation cross turning into the word either press or listen. So if the word press appeared, then participants pressed one of the 4 buttons, and the corresponding circle was filled in the screen. And after a fixed delay of a few milliseconds, a sound was presented. The sounds were exemplars of 4 different categories, which were animal sounds, human sounds, environmental sounds, and musical sounds. And there was a fixed button category. So there was a fixed mapping between the button press and the circle filled and the sound category that was played. If the word listen appeared, then one of the circles was filled without the subject's intervention, and a sound of the corresponding category was played. These two types of events we now refer to as the motor visual auditory and visual auditory, since beside the action and the sound, there is now always also the visual cue of the circle filling in both cases. Thus, in the predictable condition, there was a full predictability on whether the sound would appear, because all the presses and or all the visual cues of the circle filling were followed by a sound. When the sound would appear, because the delay between the press and or the filling of the circle was fixed, and what category the sound would be from, as there was a fixed mapping between the button press, the circle filled, and the sound category that was played. Now, these two types of events were also presented in the encoding phase of the unpredictable session. But here, there was no consistent mapping between the button that was pressed, the circle that was filled, and the sound category that was played. And additionally, the delay between the button press or the circle filling and the sound was varied randomly on every trial, chosen from 150 or 750 milliseconds. So both the sound category and the sound timing were unpredictable, both for the self-generated and for the externally generated sounds. Only 50% of the button presses resulted in a sound being presented, and only 50% of the visual cues of the circle filling resulted also in a sound being presented. So the motor visual auditory events and the visual auditory events were intermixed randomly with the motor visual and the visual only events, in which everything was the same as before, except that the sound was not played. So therefore, the sounds following an action and sounds not following an action were all equally unpredictable. In terms of whether they would appear, in terms of when they would appear, and in terms of what category would appear. For the an encoding phase of including 10 sounds, then there was a retrieval phase with 20 sounds presented. Half of them old, half of them new sounds, and of the old sounds, half of them had been encoded after an action, and half of them not. So what did we find? Once again, we examined the auditory ERPs at encoding, and we replicated the attenuation of the auditory N1 for sounds that followed an action. But here, we did not find self-generation effects for the P2. This is preliminary data, so here we have only included the sounds that were presented with the 750 millisecond delay. So this could explain the lack of the self-generation effects on the P2 component. The P2, however, was modulated by the main effect of predictability. So predictable sounds elicited an attenuated P2 component, which is in line with many previous findings of suppressed responses to predictable stimuli. There was no interaction between the effects of action and the effects of predictability. At retrieval, this time we did find a production effect. So we have better recall for sounds that were encoded following an action. But to our surprise, this effect was present both in the predictable and in the unpredictable sessions, and it was not modulated by predictability. And also there was no effect of predictability. So why is there now a production effect also without contingency? Well, this experiment differs from the previous ones in several ways that could explain this. So first of all, here we use identifiable sounds that could be named, and they were thus also easier to remember. And this was compensated with a much longer encoding and retrieval phases with many more events included. So this could be one reason. And second, if there is a negative effect of the sound action coincidence on memory, like the second experiment indicated, that the reverse production effect, then perhaps this effect only affects sounds presented in very close proximity to the action. And in experiment 2, the sounds were presented immediately without delay after the action, while here there was a delay of either 150 or 750 milliseconds between the action and the sound. So the type of sounds and the delay are additional variables that we definitely need to explore to be able to clarify this point. So in summary, I have shown data from 3 experiments with inconsistent results. Regarding our main question, whether the production effect on memory is a consequence of the known self-generation effects on sensory processing, altogether our data seems to indicate that there isn't a direct relationship between the two. While the self-generation effects remain quite constant across all the different experiments, the effects on memory performance have been null, positive, or negative in the different studies. But in any case, the results do indicate that different factors contribute to and modulate the exact shape of the self-generation effect. So perhaps we have still to find the precise factors that play a role in memory. Some of the future directions that we are taking is to to move closer to the production effect studies with designs that are more similar, and evaluating memory for self-generation effects, evaluating memory and self-generation effects for self-generated sound sequences, or to investigate in detail what the contribution of predictability is to self-generation effects by parametrically manipulating the level of contingency. Another factor that might be key is the type of sounds that are being generated, and somewhat tied to it, the intentional context in which they are generated. If you are interested in knowing more about how we are trying to pull all these threads apart and to shed some light on this, do visit the presentations of my PhD students, Nadia Paraskevudi and Marta Font Alaminos, who did all the work that I presented here. And if you also want to know how these issues relate to the sense of agency, then you can find the answers at Stephanie Sturm's presentation. Thank you for your attention.